Today I'll be showcasing my research on the potential treatment of lupus in postmenopausal mice model via polymeric nanoformulation of cyclosporin and urolithin. So first, you may ask, what is lupus? Lupus, also known as systemic lupus erythematosus, is a chronic autoimmune and inflammatory disorder that affects multiple systems and organs throughout the body. In fact, it is usually caused by an unregulated pace of antibodies surging through our lymphatic system and our bloodstream. So essentially what happens is that they cause an adverse reaction within a lot of these healthy and harmful cells due to them attaching to those. And then our white blood cells come along and cause apoptosis in those cells. This causes the cells to self-destruct, and of course this is harmful and causes inflammatory responses in our body. On a normal, healthy body, it will only be in the harmful cells, which usually happens according to our bodies. So with this anti-inflammatory response of urolithin and cyclosporin, we usually have a lot of treatments, but there is no cure. We have pharmacological treatments such as steroids like prednisone, or we have our anti-inflammatory treatments like hydrochloroquine, and we have immunosuppressants like cyclosporin. In this study, we focus on cyclosporin. It is a medical innovation, and on the left-hand side, you will see the market available Neoral. This is essentially just the market available name for cyclosporin. It is a potent immunosuppressant commonly used in organ transplantation and approved by the FDA, of course. So because it is used in organ transplantation, it suppresses our immune system from attacking these foreign objects entering our body, giving our bodies a little more time to adapt to the organs that will come inside. We have many side effects with cyclosporin, considering that it causes nephrotoxicity within our bodies and has a narrow therapeutic index. Nephrotoxicity, of course, is toxicity to our kidneys, and a narrow therapeutic index means there's a fine line between a treatment that is helpful to our bodies and a treatment that is toxic to our bodies. In a bid to overcome these side effects, the Kumar lab has come up with a new cyclosporin-laden nanoparticle that facilitates the targeted delivery of this polymer to the lymphatic system, where most of the cells that are responsible for lupin have come up with. You'll notice on the right-hand side we have cyclosporin in addition to urolithin. Cyclosporin, of course, is the immunosuppressant to combat lupus, and urolithin is actually an anti-inflammatory agent to aid in its treatment. Now, for the project activities I have identified in determining the efficacy of our polymer in this lab, first, I will begin with polymer synthesis and characterization. Second, I will go to the nanoformulation preparation and characterization. Third will be the animal study, and fourth will be the plasma and tissue processing and analysis. Now, to begin with polymer synthesis, here on the screen you will notice the polymer that we have come up within our lab. It is P2S. It has multiple functional groups. If you notice on the left-hand side, you'll see COOH on the tops and bottoms of the carbon chain. So these are actually those carboxyl functional groups, which will serve for a later purpose. As you see to the right, there is a linker called diethylamine. This is a 2-carbon linker that allows for our polymer to attach to its amine groups, the NH2s. These NH2s are the sites where our gambogic acid will attach to our polymer. And the gambogic acid is one of the most important parts of this polymer, considering that when we ingest it and it goes inside of our lymphatic system, this is what allows it to go to our intestinal wall. So the gambogic acid essentially serves as a cog on a cogwheel. Imagine that it is going through our body, and because we have more than the FDA approved version, it has more cogs to turn gears that would be the respective receptors in our stomachs. Now, you will notice that it goes through many transformations with our polymer, starting at P2S and ending up with P2S-GA, GA denoting the gambogic acid that is now attached. So once we have our P2S-GA, we start with our polymer characterization. We begin first with our NMR, or our nuclear magnetic resonance spectroscopy. This is used to identify our functional groups and make sure they have successfully binded to our polymer. Next, we go to our FTIR, or our Fourier transform infrared spectroscopy. Here on the graph, you can see that around 1,650 and 1,550 centimeters are where we have our complete conjugation and amide bonds. Third, we have our gel permutation chromatography. This is used to determine the molecular weight of our polymer. We need to know the molecular weight so that we can determine the dosage for our animals later in the study. we found that our pre-polymer was around 6,000 grams per mole and our actual polymer was around 10,000 grams per mole. Secondly, we have our nano formulation. This is where we encapsulate the cyclosporin with our particle. So we start with our P2SGA and we put it in an oil and water emulsification process. Secondly, we go through a homogenation process and leave it for some time in ethyl acetate to evaporate. And last, we go to our ultracentrifugation process, which allows the mixture to come into a pellet form. That pellet form is just the solid form of the liquid nanoparticles. We then have a resultant polymer of P2NS-GA-CSA, the N denoting that this is now our nanoparticle, GA, of course, our Gambogic acid, which is the ligand that allows it to attach inside of our intestinal wall, and the CSA is the cyclosporin, which serves as the immunosuppressant, calming down our immune system with lupus. Now, for the characterization of our new nanoparticle, we have two steps. We have our HPLC and our DLS, our high performance liquid chromatography and our dynamic light scattering. They are for determining the amount of cyclosporin encapsulated within our particle, and they are for determining the size and zeta potential of our particle. Now here you can actually see the data we retrieved from these protocols. You see, starting from the left-hand side, we have our entrapment efficiency, which is around 87%. This means that for all of the polymer that we have, around 87% has the cyclosporin actually attached. We also have the size, which is around 260 nanometers. We usually like to optimize for between 200 and 400 nanometers for our polymer to fall within, because over 400 will actually result in adverse side effects to our kidney because it cannot pass through our kidney wall at that size. And our zeta potential is around 26.6. This is essentially just the charge of our polymer. Now, third, we will have our animal study. We'll actually be working with 7 groups of animals to test our polymer on. So for our first group, we have our healthy animals. Our second group is our positive control lupus animals. Our third will be our positive control ovariectomized group. Our fourth will be treated with P2NS-GA-CSA. Our fifth will be treated with Urolithin. Our sixth will be treated with both, and our seventh will be a control group treated with both. Now, for our fourth step in this project, we'll start with analyzing the plasma and tissue of the mice models after their tenure within our project. So we have our drug estimation using LC-MS. We also have a molecular analysis of things such as RNA extraction or ELISA, and we have histological analysis like histochemistry or immunohistochemistry. And we do this with the future aims of the corresponding translational implications within our mice models to determine the efficacy of our polymer on our lupus-induced mice models. These are my references, and do you all have any questions or comments?
First of all, just wanted to say great job. Throughout your presentation and even in your title, I noticed that the absence or inclusion of ovaries plays a big part in your research. Would you be willing to explain a little bit more as to why that is?
Yes, of course. So for our mice models, we have determined that within lupus, it's very prevalent in women. So 9 in 10 of our lupus patients were actually female, or in general are female, and so we have determined that because hormones play a significant role in lupus and can be one of those factors, we're trying to see if estrogen produced by the ovaries has any implications in our study as well.
How do you find the mice that have lupus? Are you inducing it, or is there a certain protocol that you use to find them?
So lupus can be induced. However, within our particular breed of mice, which I'll go back to our animal study, you'll see that they are MRL-LPR mice. And so that means that they're actually bred with lupus themselves. So essentially within this selective breeding group, we choose the ones that have already been born with it, and then we also get the ones that are healthy and kind of do our tests on our subjects.
How do you encapsulate the polymers at such a microscopic scale?
Got it. So for our nano formulation, this is where we use our oil and water emulsification process, and we will actually have our polymer and our oil together, and we will spin it at very high speeds, around 5,500 revolutions per minute, and this allows it to come together and homogenize.